Good afternoon, everybody. On behalf of the organizing committee of the Immuno webinars, which is a joint effort of the University of São Paulo and the Federal University of Rio de Janeiro, we are very pleased, and it's really an honor to receive Dr. Dwyer for his talk on the role of TGF-b in the regulation of intraepithelial mast cells development and activation. In another installment of this Immuno webinar series that happens every Monday at noon on this Zoom room and the YouTube channel of Immuno webinars. Dr. Dwyer got his bachelor's in biology at Amherst College and his PhD on immunology at Harvard, which was followed by a period as an instructor of medicine at Brigham and Women's Hospital, where he is currently an assistant professor of medicine. Dr. Dwyer is one of the most prominent young researchers on mast cell biology, with many contributions to the field, such as the landmark 2016 paper in Nature Immunology on the expression profile of constitutive mast cells in different tissues, and was followed by seminal work on mast cell differentiation and function under different inflammatory conditions, working with names such as Nora Barrett, Josh Boyce, and also Frank Olsen, who also had the honor to work as a postdoc researcher in the beginning of 2000. Dan is really one of the nice guys. Very hardworking. I had the chance to work by his side when I was spending a sabbatical year at Harvard, and I'm sure you guys will have a great time and learn a lot about mast cells with his talk. Dan, thank you very much. The floor is yours.
Thank you. Great. So thank you so much, Bruno, for the opportunity to come and talk to you all about some of the work that my lab has been doing recently. So mast cells start their life as this pretty innocuous circulating progenitor. It is not something that on a slide you could really pick out. And it's really not until they actually reach their target tissues that they take on this really distinct morphology that's classically associated with mast cells, where they become very filled with these granules that have this metachromatic feature when stained with certain histologic stains. And my lab studies this transition from the fairly featureless agranular progenitor to this very distinct differentiated effector cell within the tissue, and specifically trying to understand the tissue signals that instruct progenitors during this transition. Now, once mast cells mature, they are able to respond to a wide range of activating signals and influence inflammation through a number of discrete pro-inflammatory pathways, which is nicely illustrated by this figure here from a review by Steve Galley. And depending on the paper you read about mast cells, they can respond to and produce just about every inflammation associated factor that you can think of. But believe it or not, this figure here is also a bit of an oversimplification. So we know, for example, that not all mast cells express all of the receptors that are associated with them. So for example, MRGPRX2, which is the receptor for substance P and various xenobiotic compounds, and CD88, which is the receptor for C5 component of complement, are highly expressed by mast cells in the skin, but only weakly expressed in the lung, where mast cells in the skin tend to have much lower Fc-epsilon-R1 expression. So clearly there's a lot more heterogeneity to the cell than just mast cell. So instead, so in humans, we generally classify mast cells into several histochemically recognized subsets that reside in discrete tissue microenvironments. So mast cells in the skin at steady state contain the proteases tryptase, chymase, carboxypeptidase A3, and cathepsin G, so termed MCTCs because they have both tryptase and chymase, whereas the mast cells that are in the epithelium are generally referred to as MCT because they really just contain tryptase, at least at steady state. We know that the granule components are a little bit different between these. So mast cells in the skin have a lot of heparin in their granules, whereas those in the epithelium are more chondroitin sulfate. We know that there are receptors that are associated with mast cells in the skin that we don't see elsewhere, such as CD88, the receptor for complement, and MRGPRX2. And we also think of mast cells within mucosal surfaces as being much more dynamic and replenished by circulating mast cell progenitors as they are shed out into the lumen of whatever tissue they're in contrast to those in the skin, which, as best we can tell, seem to stick around for a very long period of time. Now, although the MCT-MCTC distinction is more useful, it is still somewhat of an oversimplification. So, for example, we know that in humans, mast cell distribution and phenotype is actually still quite dynamic, and it changes a lot during type 2 inflammation. So a few examples of this are that MCTCs, so mast cells containing both tryptase and chymase, start to appear in new locations during inflammation. So in severe asthma, these MCTCs start to appear in the airway smooth muscle. They can actually be found in the epithelium in severe asthma as well. And in nasal polyps, they start to pop up in the glandular epithelium. The intraepithelial mast cells, the MCT, also change during inflammation. So there is a dynamic expansion of them. They tend to very dramatically increase concentration during type 2 inflammation. And they also change their protease phenotype. So in steady state, MCT really just have tryptase. In type 2 inflammation, they'll also have this additional protease, carboxypeptidase A3. And this has been seen across mucosal tissues. It's true in the asthma or asthmatic epithelial mast cells, the mast cells in the epithelium of nasal polyps, as well as in the esophagus in eosinophilic esophagitis. So this seems to be sort of a broad feature of mast cells during type 2 inflammation. So the project I'm going to talk about today is going to start in the mouse, which is a particularly useful model to study intraepithelial mast cell development. And this is due primarily to the fact that there are two inducible proteases in mouse that are restricted to the epithelium. So as far as we know, in humans, there's no specific protease that comes up when a mast cell develops in the epithelium. But in mice, there are two proteases that come up, and they are referred to as MMCP1 and MMCP2 for mouse mast cell protease. And this has been primarily studied in BALB/C mice, where it's really easy to induce the expansion of mast cells. Well, it's somewhat labor intensive, but they expand very dramatically. So some of the most common ways people induce mast cell expansion is to use the OVA model of airway inflammation. So if mice are sensitized intraperitoneally with OVA and alum and then sequentially challenged with aerosolized OVA, there is a dramatic expansion of mast cells in the lung of BALB/C mice. So the mast cell progenitors peak first, very early on. There's a wave of progenitors that are recruited to the tissue. And then as time goes on, those mast cells will start to mature and take on this, both the histologic and protease phenotype of a mucosal mast cell. And so what we're trying to understand in my lab, and some of the key questions I'm going to go through today, are first, understanding what are the signals that direct the development of mast cells from being a progenitor to being a mature mast cell that has this intraepithelial mast cell phenotype. Secondly, we want to understand if the signals that drive intraepithelial mast cell differentiation in mouse and human are the same. And finally, we want to understand how mast cells that are within the epithelium functionally differ from those that are found elsewhere within the body. So the first project I'm going to be talking about actually started during Bruno's sabbatical at Brigham, when we were looking to establish a couple things. First, we wanted a model that we could use to expand airway mast cells in C57 mice, which is a lot less simple than in BALB/C. They're much more resistant to developing type 2 inflammation. And secondly, we wanted to find some markers that we could use to pull apart the recruited mast cell population from those that were present at baseline. So here I'm showing you flow cytometry of mast cells in the lung of a mice, and we pre-gated here on CD45 positive lineage negative cells. So when we look in a saline challenged mouse and we look at FC-epsilon-R1 versus CD117, there are really very few mast cells that are there. If we use integrin beta-7, which is previously been described as an integrin that is necessary for recruiting mast cells to mucosal tissues, we can see that beta-7 integrin versus side scatter actually fractionates this population, and we can see that the minority of the cells that are the mast cells in a saline challenged mouse have beta-7 integrin. Most of them are higher on side scatter and do not have beta-7 integrin. When we look at a mouse that has been challenged with an allergen, and here we're using two different models. The first is sequential challenge with Dwyer, a house dust mite extract, and the second is resensitization challenge with a mold extract, Alternaria alternata. In both of these models, there is a expansion of the mast cell compartments. We see there are more mast cells within the lung. And then when we use beta-7 versus side scatter, we can see that those increased mast cells all have beta-7 integrin on their surface. And when we quantify that, the beta-7 low mast cells in both dust mite and Alternaria really don't change in concentration in response to inflammation, whereas the beta-7 high mast cells do. There's the increase is very selectively of these beta-7 high mast cells in both models. So we thought that maybe beta-7 integrin could be a marker that we could then use to distinguish the recruited mast cell in the lung from those that are there at steady state. So when we sort these mast cells out using flow to look at what they histochemically look like, the beta-7 low mast cells look like a very characteristic mast cell. They have beautiful metachromatic granules. If we do chloroacetate esterase staining, which stains the protease, we can see that, again, we get this nice granular stain in them. The beta-7 high mast cells, in contrast, and this is sorted out of a challenged lung, they're very heterogeneous in their appearance. Some of them have many granules, some of them have no granules, which suggests there's a real range of maturation in this compartment. And when we do CAE staining, we can see that some of them have almost no staining, whereas some of them are more, there's more distinct staining. So to better understand what differentiated these cells and how they might change during inflammation, we turned to using RNA sequencing. And so we sorted out beta-7 high and beta-7 low mast cells from the lungs of mice, either following saline challenge or following dust mite challenge. And so when we did that, we could see that on principal component analysis, these mast cells did separate. The beta-7 low mast cells in purple for the saline and blue for dust mite were on one side and in PC1, which had the majority of the variants, and then PC2 was separated by the beta-7 high mast cells in either saline in green or dust mite challenge in red. So the first thing we did, because these are our mast cells and they're classically distinguished by proteases, is look at the protease expression profile. So we could see that the beta-7 low mast cells expressed a number of proteases that are associated with the connective tissue mast cell, including CPA3, kinase, various tryptases. Whereas the beta-7 high mast cells had these mucosal mast cell proteases in them, MCPT1 and 2. And interestingly, they also had MCPT8, which is a protease that's mostly associated with basophils and actually used to drive basophil deletion in a couple models. So when we then look at protease components, we can see that the beta-7 low mast cells have an expression of a number of enzymes associated with heparin biosynthesis, whereas the beta-7 highs had CHSY1, which is chondroitin sulfate synthase. So this suggests to us that the beta-7 high mast cells were going to be the mucosal phenotype of mast cells. So to confirm this, Mina Derrickson, who is extremely fortunate to recruit as my first postdoc, and she did pretty much all the work I'm going to be showing you today, was able to develop a staining protocol to use flow to look at intracellular MCPT1 expression. And when she did that, we could see very nicely that all of the MMCP1 was within this beta-7 high compartment. So this was the mucosal mast cell compartment in these B6 mice. So next, we wanted to understand what happens to these cells, these recruited mast cells during airway inflammation. And there actually weren't a huge number of genes that changed. And this is likely due in part to the fact that we didn't have a great sample size, and also that dust mite is not a extremely immunogenic compound, at least the Dwyer that we used. Since then, here we've developed much stronger protocols to recruit mast cells. So we saw there were a number of genes that changed. These were largely upregulated. And there were some interesting things. So for example, they upregulate TPH1, which is involved in dopamine biosynthesis and various other enzymes associated with dopamine. They also upregulated a number of inflammation-associated transcripts, including the chemokine CCL1, a couple IL-6 family members, IL-6 and LIF, various lipid mediator biosynthetic enzymes, including COX-1 and thromboxane synthase, as well as a number of activating receptors, such as the IL-33 receptor, IL-1RL1, and FCS1R1, the IgE receptor. So even though there weren't a huge number of transcripts that were changing here, it did look like they were somewhat important ones that were being upregulated. So in the mouse, many signals have been proposed to direct this intraepithelial mast cell phenotype, and that includes IL-10, IL-9, TGF-beta, and Notch signaling. So we hope that we could maybe use our in vivo data set to help understand which of these signals might be playing a role in driving the development of these mast cells. So when we looked at the individual genes a little bit closer, we noticed that a number of them had been previously identified as TGF-beta target genes, either in mast cells or in other cell types. And this included the mast cell protease MCPT1 and 2, alpha E integrin, which has been shown to be a downstream of TGF-beta in multiple cell types, as well as SKIL, which is actually induced by TGF-beta and negatively regulates the pathway, and it seems to be very strongly upregulated by TGF-beta signaling. So we wanted to confirm that TGF-beta in our hands could induce MMCP1. So we took bone marrow-derived mast cells and we treated them with TGF-beta. And we were actually a little disappointed to see that we had very, very poor induction of MCPT1 and alpha E integrin. But then when Mina added stem cell factor in addition to TGF-beta, then we saw there was a much more dramatic upregulation of MMCP1. Instead of just a few percent of the cells upregulating it, essentially all of the cells upregulated it. We could also see there was stronger upregulation of alpha integrin or alpha E integrin. So I think this told us, this sort of suggested to us that really what these cells need to turn on MCPP1 is sort of a two-hit system. They need to both see stem cell factor and also see TGF-beta. And then all that upregulation is quantified here. They all dose response, and Bruno can tell you one of the things you learn in Frank Austin's lab is if it doesn't dose response, it is not science. So we always check to make sure everything can dose response. So we then wanted to understand beyond just those few transcripts we picked up or we plucked out, the degree to which TGF-beta was actually regulating the beta-7 high mast cell transcriptome. So to understand this, we took BMMCs and we treated them with either stem cell factor, TGF-beta1, or both of them together. And we did RNA sequencing. And we saw there were a number of transcripts that were differentially regulated. And looking at the pattern, we could see that there was this interesting cassette here that was, or there were a few interesting cassettes of genes, some of which were induced by stem cell factor and enhanced by TGF-beta, some of which went in the other direction, some that were induced by TGF-beta and enhanced by stem cell factor. So we started to then dive into those transcripts. We can see that a number of them were associated with the granule. So notably, the combination of stem cell factor and TGF-beta downregulated NDST2, which is important for making heparin, upregulated chondroitin sulfate synthase, as well as MCPT1 and 2. We can see that it turned on a number of inflammatory mediators that were upregulated in our beta-7 high mast cells. This included IL-6, COX-1, and DDC. Notably, it downregulated a number of MAS-related G-protein coupled receptors, although there was one that was upregulated. And in general, when we compared the transcriptomes, we could see that many of the transcripts that were upregulated in beta-7 high mast cells in vivo were in fact downstream of our TGF-beta. So we used this data set to then construct a gene list that we were able to use to probe our in vivo data set using gene set enrichment analysis. We see that there was a highly significant enrichment for this TGF-beta score in mast cells in vivo, which told us that this was likely, if not TGF-beta itself, something downstream of that. So this SMAD signaling was likely very important for driving the intraepithelial mast cell phenotype in mouse. So we then wanted to understand where this signal was coming from. And so when we look at this is looking at epithelial cells in the lung of the mouse. So we hypothesized that the TGF-beta signal could be coming from the epithelium because there was actually a lot of literature support for the fact that this is an epithelial-associated signal, even though it's, as immunologists, we often think of it as being a regulatory T-cell anti-inflammatory cytokine. So we saw that when we looked at epithelial cells, they all expressed TGF-beta for the most part, although this didn't really change much in response to dust mite. We could also see that when we cultured epithelial cells in vitro, there was nice expression of TGF-beta on their surface. So Mina then did the experiment where she took BMMCs and she co-cultured them with epithelial cells for about 1 week. And when she did this, she saw that there was a very nice induction of or of MMCP1 on the surface of these cells. That was also true for alpha E integrin. So then Mina did the critical experiment where she used a TGF-beta receptor inhibitor, LY21097661. And what she was able to find was that when we co-culture these BMMCs with epithelial cells, and we're not adding anything else here, just a little bit of IL-3 to support the viability of the mast cells, there was this very dose-dependent inhibition of the MMCP1 and alpha E upregulation when she used this TGF-beta receptor inhibitor. So we haven't yet done the critical in vivo experiment to close the loop here. That's going to be the focus of some future work. But from this, we were able to, I think, pretty strongly conclude that it is very likely that epithelial-associated TGF-beta is a critical signal in driving this induction of the intraepithelial mast cell phenotype in mouse. Which then brought us to the very obvious question of, could TGF-beta be playing a similar role in regulating the human intraepithelial mast cell phenotype? And there is some reason to expect that this might be the case. So there is an interesting association between TGF-beta signaling and asthma. There are SNPs that are associated with either TGF-beta itself, the TGF-beta receptor, or SMAD proteins that have been identified in several different GWAS studies as being associated with the development of asthma. And there was a very nice paper that was published in Immunity a few years ago from an Australian group, and they looked at the chromatin state of human mast cells. And they noticed that there was this interesting locus in SMAD3 that appeared to be very open in human mast cells from the tonsil, as well as ones that are grown in vitro. And this right in the middle of this open chromatin region is one of the GWAS SNPs that tends to be associated with the development of asthma. So they hypothesized in this study that there might be a link between this specific SNP and the mast cell and the development of asthma. So the model that we use to understand human mast cells is, well, I use model here, but very, very loosely, is nasal polyposis. So human nasal polyps share many of the type 2 inflammatory features that are seen in human asthma. They have mast cells in the epithelium that express CPA3. They have MCTCs that expand in strange places. But importantly, in contrast to the bronchus of an asthmatic, nasal polyps are routinely surgically resected to help the patients. So this greatly improves patient symptoms, and it also gives us a very rich source of tissue to study mast cells. This is an extremely rich source of mast cells. So we have used nasal polyps to understand human type 2 inflammation in mast cells through a series of studies that I did during my postdoc. And going back into those data sets that we had previously published, Mina went and looked to see where she could see TGF-beta expression. So this was from the 2018 nasal polyposis study that we published. And when we look at that data set and map TGF-beta 1 and TGF-beta 2 onto it, we can see that TGF-beta expression is actually largely found in basal epithelial cells, both TGF-beta 1 and 2. And notably, basal epithelial cells are the cells that tend to sit right next to mast cells within a nasal polyp. And then when we looked at mast cells, or sorry, not mast cells, basal epithelial cells that we had sorted out of inflamed sinus tissue and for RNA sequencing, we could see that there was a significant increase in both TGF-beta1 and TGF-beta2 expression in epithelial cells that are from nasal polyps relative to those from non-polyp, which suggests to us that there's probably more of a TGF-beta signal present in the epithelium in these nasal polyps. So more recently, we published a single cell sequencing study looking at the various mast cell phenotypes that are present in nasal polyposis. So Mina went back to that data set and she looked to see whether she could see any hints of TGF-beta being present there. So just to orient you, we have 4 different populations of mast cells that we're able to describe in the nasal polyp in this study. The MC1 in blue express high levels of chimeras and cathepsin Gs. This would be the human MCTC. In red here is the MC3, which expressed a number of transcripts that we found to be linked to the intraepithelial mast cell. This includes the IL-25 receptor, IL-17RB, as well as GPR183, which encodes EPI2. We saw this interesting population in the middle that seemed to be at an intermediate point. We proposed that these are transitional mast cells that are earlier in development, and they're still choosing what direction they're going to differentiate into. And then we also had identified in that study that there's this proliferating population of mast cells that is present in the nasal polyp, which we think probably plays at least some role in the mast cell expansion that we see. So when Mina looked at the gene lists that we had constructed in this study, she was able to see that TGF-beta target transcript SKIL was quite significantly increased in the MCT in nasal polyposis relative to the MCTC, which hinted that there might be a TGF-beta signal that was present in the intraepithelial mast cells in humans as well. So to understand that better, Mina decided to construct a new single cell sequencing data set using more recent technologies. The sequel study that we constructed was, by this point, the sequencing technology was several years out of date. So she sorted nasal or mast cells from the nasal polyp of 4 aspirin sensitive donors. We sequenced them using the 10x Genomics B3 Chromium platform. And then when we looked at the various clusters, we could see through just some control observations, I'm not going to get into it for the sake of time, that we had 2 mast cell, 2 MCT populations. So these would be intraepithelial mast cells, one of which seemed to be more activated and one more quiescent. We had our intermediate transitional population. We had a cluster of MCTCs. And again, we had that proliferating population present. So Mina then mapped on the murine TGF-beta target transcripts that she had identified in the mouse study. And we could see that there was a pretty striking enrichment of these transcripts in the intraepithelial mast cell compartment. And in addition to SKIL, which we had previously noticed, there was SMAD7, which is another TGF-beta regulatory part of the TGF-beta regulatory pathway, several transcription factors, alpha-E integrin, chondroitin sulfate synthase. So together, this suggested that maybe TGF-beta was playing a similar role in directing the human intraepithelial mast cell differentiation. So to understand this better, Mina took CD34-derived mast cells. And so we're taking human peripheral blood. We're using CD34 beads to pull out the progenitor pool of the blood. And then growing them under mast cell differentiation conditions. In human, that means stem cell factor and IL-6 for 6 to 7 weeks until they reach maturation. So she did this for 3 donors and then treated them for 7 days with either vehicle or TGF-beta, after which we sent these samples off for RNA sequencing. And we could see that there was a pretty substantial impact of TGF-beta on the intraepithelial mast cell transcriptome. There were about 1,000 genes that were upregulated and even more that were downregulated over the course of these 7 days. And something that really jumped out at us was amongst these most differentially regulated transcripts, there was a really profound downregulation of these MCTC-associated proteases, chimase and cathepsin G, as well as MRGPRX2, which is similarly associated with MCTCs. So Mina then used these differentially expressed genes, which I'm going to get into a little bit more later on in the talk, to construct signatures that she could use to probe her single cell sequencing data set. And when she did this, we could see that the top 200 TGF-beta upregulated genes were most enriched in the MCT1 and 2 clusters and lowest in the MCTC. And in contrast, the TGF-beta downregulated genes, so this is the things like cathepsin G and kinase, were highest in the MCTC and lowest in the MCTs. Here, though that is displayed as violin plots, what we're showing here in these violin plots is the percentage of the transcriptome of each single cell that are these top 200 TGF-beta upregulated or downregulated transcripts. So, the percentages here are decently high. We can see that there's actually a very strong connection between this TGF-beta signature and what the cell is actually expressing. So before diving deeper into the data set, we wanted to understand if we were looking at something that was restricted to nasal polyps or more broadly associated with intraepithelial mast cells. So Mina pulled 2 additional single cell sequencing data sets that have been generated by others and deposited online. The first is from healthy distal lung, and this is the study that was from. The second was from ulcerative colitis, and here's the study that was from here. So in both of these tissues, we could find clusters. Well, I'll start with the distal lung. We can see that there was a cluster that was had virtually no kinase cathepsin G in yellow, and that was the one that had the highest expression of this TGF-beta transcript score, although the differences were not as large as we saw in the nasal polyp. And then when we looked at ulcerative colitis, there was a cluster there that was predominantly made up of mast cells from the epithelium. So in this study, they actually had fractionated the epithelium and done single cell sequencing. So that cluster that was really associated with the epithelium had, again, the highest TGF-beta expression score in it. So this suggested to us that this is going to be a part of a larger program that's regulating intraepithelial mast cell phenotypes. So now getting into some of those differentially regulated genes. So as we had seen in mouse, many of the differentially expressed genes in these peripheral blood-derived mast cells were associated with proteases. So we saw there was upregulation of transcript encoding tryptase and CPA3 following TGF-beta treatment, which actually, I should say, does match differences in expression we do see in vivo, as well as downregulation of cathepsin G, kinase up here, NDST2, which is going to be that first enzyme in making heparan sulfate. But when Mina then did intracellular protease staining on her peripheral blood-derived mast cells, she saw that there was really no difference whatsoever in the kinase expression over the 7-day window. So we know that mast cell granules are incredibly stable, and they were held in this almost crystalline state prior to degranulation. So we hypothesized that maybe this was because the mast cell granule was so stable, we weren't really seeing differences in protease expression. So Mina then came up with the experiment of taking CD34 cells and growing them under mast cell differentiating conditions, either with or without TGF-beta for the full duration. And when she did this, she could very nicely see that there was this distinction between mast cells grown in each phenotype. So mast cells grown in TGF-beta in the blue had markedly reduced kinase expression intracellularly compared to those treated with vehicle in blue, TGF-beta in the red and the vehicle in the blue. That was true for both kinase and cathepsin G. And we could also see that there was a little bit down regulation of CD117 by TGF-beta, which is consistent with what other people have shown in literature in the past. Now, an interesting thing that she found was that it appears that this inhibition of kinase requires continued TGF-beta signaling. So if Mina takes her mast cells that have been grown in TGF-beta and removes TGF-beta for 2 weeks, there is a very marked upregulation of kinase, which might have some implications for diseases where MCTCs appear in the epithelium, such as severe asthma. And this is not just true for mast cells that are grown and developed in vitro. Mina also was able to sort MCT and MCTC from nasal polyps and culture them with either TGF-beta or vehicle for 2 weeks. And when she did that, she saw that MCTCs that are cultured in TGF-beta maintain a very kinase low phenotype, whereas those that are treated with that do not have TGF-beta in the media start to upregulate kinase, and that's quantified here. In contrast, MCTCs that are sorted from in vivo are highly stable, much like the ones we saw in vitro. So there is really no impact whatsoever of being cultured in the presence of TGF-beta. So this tells us that there is this interesting one-way plasticity with human mast cells, as far as at least the granule protease content goes, where mast cells that are that have kinase, they will not lose that kinase, or at least culturing them under MCT conditions does not reduce kinase expression, even though transcriptionally they change to look much more like an MCT. Whereas in contrast, when we take an MCT and we remove the TGF-beta signal, there is this marked upregulation of kinase, and that's true both in vitro and with in vivo derived mast cells. So next, we wanted to get into trying to understand, use these data sets to understand some of the differences in effector roles for MCT and MCTC, so the intraepithelial and subepithelial mast cells in humans. So we do a direct comparison of the MCTC, and we're comparing that here to the red MCT cluster, which is the more activated cluster. We could see that there was marked differential expression of a number of transcripts that suggest that they are going to have really discrete contribution to host inflammation. So going beyond the proteases that have been previously mentioned, although I should say one of the things that we saw that was kind of interesting in the MCT that we're following up on is that there are a number of proteases that are induced in these cells relative to the MCTC. So it might actually be that the intraepithelial mast cell phenotype is a bit more rich and then like the mouse than has been previously established. We also saw they have higher expression of tryptase and CPA3, which again, we had seen to be targets of TGF-beta in vitro. There's differential expression of a number of cytokines and chemokines and growth factors, the MCTC being very enriched for chemokines. They have a lot more IL-13 expression, as well as LIF, which is an IL-6 family member, whereas the MCT, although they had lower IL-13, they had higher levels of IL-5, which is a sort of different type 2 cytokine or type 2 inflammation-associated cytokine. And usually we think of IL-5 and IL-13 as really going hand in hand, but this suggested we actually had some separation between the two. When we looked at lipid mediator biosynthetic enzymes, higher expression of COX-2 and PGD synthase in the MCTC, whereas the MCT had higher expression of COX-1, which we had actually also seen in the mouse intraepithelial mast cell, as well as a number of components of the cystidoleucotriene biosynthetic pathway, including ALOX5AP. We could also see, although I won't really get into these too much, there was a lot of differential expression of various receptors for cytokines and chemokines, as well as for activating and inhibitory receptors. So this also suggests that the way that MCTs and MCTCs see the world around them is going to be quite different. So one of these differentially expressed sensing transcripts was MRGPRX2. Now, as I mentioned very, very briefly at the beginning, MRGPRX2 has kind of been well studied recently in mast cells. It is the receptor for substance P, which is a neuron-derived peptide that is very good at activating mast cells. Also compound 4880, which has been used for many decades to study mast cell activation, acts through this MRGPRX2. So we could see that after 7 days, even though, again, I'll remind you that the kinase did not go away, but the MRGPRX2 expression was markedly decreased by flow cytometry after 7 days. And consistent with that, when we took these mast cells, either grown with or without TGF-beta, and we treated them with compound 4880 or substance P, we could see that there was a very significant difference in how those mast cells were activated. When mast cells were grown in TGF-beta for the full duration of culture, we could see that there was an even stronger difference in MRGPRX2 expression. And consistent with that, it was virtually impossible to activate these TGF-beta grown mast cells with compound 4880. So even though TGF-beta did have some impact on transcripts associated with Fc epsilon R1, and those are shown here on the left. There was really no difference in expression levels that we noticed by flow. If anything, there was slightly more TGF-beta expression on the, or sorry, slightly more Fc-epsilon-R1 expression on the TGF-beta treated cells. And there was no difference in their ability to granulate, to degranulate following IgE cross-linking, which told us that we then had a tool that we could use, namely activating these cells through IgE, to start to probe their effector function and to see if they had any sort of differential capacity to make mediators in response to activation. So first, I'm showing you cytokine and chemokine production. So I will remind you that one of the differences that we saw between the MCT and the MCTC in vivo was that the MCT seemed to have more IL-5 production, the MCTC more IL-13. So if we take mast cells that are treated with TGF-beta, we can see that in response to both IgE and IL-33, there is an increase in IL-5 production in these TGF-beta treated cells. That's highly significant for IL-33, much less of a trend with IgE, although one of the things that we weren't necessarily expecting here was that we found there's a huge difference in magnitude of these mediators between IgE and IL-33. So much more cytokinergic IL-33 signaling. In contrast, following IgE cross-linking, we saw a trend towards a decrease in IL-13 in the TGF-beta treated cells, and that was significant in our IL-33 treated cells. So this tells us, looking at this together with the differences in sequencing we saw in vivo, that TGF-beta, rather than being broadly anti-inflammatory on these mast cells, does appear to instead be shaping how they respond to activating stimuli. Then lastly, we looked at CCL2. There was really no difference in CCL2 following IgE cross-linking between the two, but a significant decrease in CCL2 in our TGF-beta treated mast cells. And this is now showing expression of those mediators in MCTC and MCT in vivo. Much like we saw in vitro, CCL2 is lower in the MCTC, CCL2 is higher in the MCTC. Lower in the intraepithelial mast cell. IL-13 shows the same pattern, whereas IL-5 is significantly increased in the intraepithelial mast cells. So we also noticed that there was a pretty strong impact of TGF-beta signaling on lipid mediator associated enzymes. Notably, the cystinonucleotide associated enzymes, ALOX5AP, LTC4 synthase, and ALOX5, which were all upregulated by TGF-beta. And COX-1, which is associated more with prostaglandin synthesis, also upregulated. And that largely mirrored differences that we saw between the MCT and MCTC in vivo, where in our nasal polyp mast cells, the intraepithelial mast cell had higher expression of ALOX5AP and COX-1, whereas the MCTC had a higher expression of COX-2 and PhD synthase, although neither of those were differentially regulated by TGF-beta. So we wanted to understand if this was going to then change their capacity to make lipid mediators. And we saw that the TGF-beta treatment actually had a very profound impact on the ability of mast cells to make lipid mediators. So there was a significant enhancement in cystinonucleotide production by mast cells following TGF-beta treatment. We saw very little expression or little production in mast cells activated through IgE under our standard growth conditions, whereas there was a very significant increase following TGF-beta treatment. And we saw there was similarly an enhancement in the ability of these cells to make PGD2. PGD2 was induced by IgE cross-linking in both conditions, but substantially increased in mast cells that were treated with TGF-beta. And that is not just an in vitro phenomenon. So Mina, again, was able to sort mast cells out from nasal polyps. She sorted out MCTs and MCTCs, and then she activated them with IgE cross-linking or left them unstimulated. And when we looked at cystinonucleotrides, we could see that there was a pretty profoundly increased capacity of the intraepithelial MCT to make cystinonucleotrides, although there was an increase following IgE cross-linking in both compartments. On a per cell basis, the MCTs were much better at producing cystinonucleotrides. Although this was not statistically significant, there was also a trend towards being an increase in their ability to make PGD2, again, largely mirroring what we saw in vitro. So the PGD2 was interesting to us because this is something that's largely associated in literature with the connective tissue mast cell in mouse, or the subepithelial mast cell, the MCTC in humans. So we hypothesized that this increase in PGD2 could actually maybe be downstream of this COX-1 upregulation that Mina noted, which, again, kind of bringing things full circle, was also one of the more highly upregulated transcripts in the mouse intraepithelial mast cell in response to both TGF-beta treatment and in vivo following dust mite. So Mina then tested this using two inhibitors. SC560 is a COX-1 specific inhibitor, so PTGS1, and then SC236 is a COX-2 specific inhibitor, PTGS2. So we found that actually in both the the mast cells that were treated with or without TGF-beta, it seemed like the PGD2 production was really largely downstream of COX-1. So there was a significant decrease in PGD2 production in both types following inhibition of COX-1. And it really brought them down to essentially the same level. And I should say the difference between the two conditions here was significant, although we don't have the stars drawn in here. In contrast, COX-2 really had minimal impact on PGD2 production in either cell type. There was a slightly significant downregulation in the TGF-beta treated cells. There was really a slight trend, but no significance in the non-TGF-beta ones. So this suggested to us that probably it's that PTGS1 or that COX-1, COX-2 is really the rate limiting step in PGD2 production. And so by increasing COX-1 levels, TGF-beta primes these cells to be able to make much more PGD2 following activation. And it looks like this may be also true in vivo. So Mina was able to do intracellular staining for COX-1 in our previously defined MCTC, MCT, and transitional mast cell gates. So again, the MCTs are the intraepithelial mast cells. And she saw there was a significant increase in COX-1 expression in both the MCT and the transitional mast cells relative to the MCTCs, which actually would suggest that maybe these transitional mast cells have some effector capacity too. So in conclusion, environmental signals really do seem to play a major role in shaping mast cell responses to activation. And we think that SMAD signaling, potentially through TGF-beta, although there are other ways you can get to SMAD signaling, such as activins and BMPs, which these mast cells also express receptors for, likely is a central contributor to the intraepithelial mast cell phenotype in both mouse and human. Functional distinctions between mast cell subsets go far beyond their protease expression. Even though that is histologically the easiest way to tell them apart, they're probably playing very, very different roles in contributing to host inflammation. And finally, mast cells execute discrete transcriptional programs that are directed by their tissue microenvironment. So the intraepithelial mast cells we found had increased capacity to make IL-5, decreased ability to make IL-13, and various chemokines. And the intraepithelial mast cells were had greatly enhanced lipid mediator production capacity, which is interesting to think about in diseases that are associated with increased lipid mediators systemically. And in fact, aspirin-sensitive nasal polyposis is one of these diseases, as well as asthma, where there's increased levels of lipid mediators. So with that, I would like to thank all of the people that helped with this study. So the work here was almost entirely done by Mina Derekshan, who, as I mentioned before, I was extremely fortunate to recruit as my first postdoc in my independent lab. Eleanor Hollers is a research associate who more recently joined us and has really helped us finish this project. My whole team here at Brigham has been absolutely fantastic in helping me launch my independent career. Josh Boyce, Nora Barrett, who I did my postdoc with, Tanya Laidlaw, Katie Buckite, and their clinical team. And then the surgeons that we work with are also absolutely fantastic. Also like to thank my funding. And again, a big thanks to Bruno for this opportunity. This was really fun.
All right, then. As expected, very nice talk, lots of data. and very interesting and important data. And we, during this webinar series, what we have been doing is to open for questions from the students before anybody else. So I have a question from one of our students here.
Hi, Professor. Can you listen to me?
Yes.
Yeah.
Okay. So my question is related to COVID-19. As you talked a lot about airway cells and these inflammations, and I would like to know if your group has any study correlating TGF and its effect in mast cells during COVID-19.
So no, we have not looked at anything related to COVID-19. It's an interesting, topic though. One of the things that sort of jumped out at me early on is that the protease that's primarily associated with activating COVID, the TMPRSS2, it actually has some similarities in its cleavage site to mast cell tryptase. So there could be a link there. On the other hand, there's some clinical data that seems to indicate that sort of in contrast to many diseases, that having a mast cell burden is very dangerous if you're infected virally, such as influenza or mastocytosis. That appears to be much less the case for COVID-19. So these patients are not getting the severe responses to COVID-19 they might to influenza. So, I think that it is a very interesting topic. I would love to understand it more, but we've not looked into it at all.
Okay, thank you.
We have another question here. Lucas?
Hello, Professor. Thank you for the amazing presentation. It's a really interesting work. I actually have two questions. My first one is, do the TGF-beta 1 somehow induce the expression of prothrombotic factors, such as platelet activating factor or aparinase?
We haven't measured them, but it's very possible. So one of the differentially, there, a couple of the enzymes, this was specifically in the mouse, we saw that thromboxane synthase was upregulated by TGF-beta and was enhanced in these in the intraepithelial mast cells. Thromboxane is definitely something that can activate platelets. And then LPCAT2 is a gene that is really central to generating platelet activating factor, and that one was also upregulated. So certainly possible. We have not tested it yet, but transcriptionally it does suggest that they are going to have increased ability to activate platelets through these lipid mediators.
My second question is, this model of detecting and comprehending which type of cells, the infiltrating ones, which are more naive, and the resident ones that are more mature. Do you think you can apply this model to any other kind of diseases, such as other inflammatory diseases? You have a really focused attention on inflammation derived from airway allergies, but I have an interest in cancer, for example, and we have a big homing of mast cells to these tissues. So
yeah.
my question is like,
no, it's a great question. Yes, there is certainly, we've looked at a couple different subcutaneous tumor models, and there is a very impressive recruitment of mast cells to these. Beta-7 integrin does not appear to be maintained. Well, I guess I should back that up. We're not sure what the source of the mast cells are. When we look at beta-7 integrin expression, it's only on a small fraction of the mast cells that are present within the tumor. So there's probably more to it. It might be that when we're looking at the airway, so one of the things that is upregulated, right, is alpha E integrin, and alpha E beta 7 is important for binding onto, I believe, E-cadherin. So that could be a way that the cells are anchoring themselves in the airway epithelium. It might be that there are other signals that are present with other cells that are going to push them in other directions. So yeah, when we have looked at, we've only done sequencing on colon adenocarcinoma subcutaneously. They did appear to turn on many transcripts associated with intraepithelial mast cells, but also many associated with more connective tissue mast cells. So probably, I think one of the things that we're hoping to explore more going forward is it really seems like the mast cell phenotype is not, it's about how they're integrating many different signals that they're receiving from their environment rather than there being kind of one master program. So we think that TGF-beta is one layer of the intraepithelial mast cell phenotype, but we know when we look at nasal polyp mast cells, there's much more to the story than just TGF-beta. So for example, IL-4 is likely playing a role. Signals like IL-33 are probably playing a role. And I'd imagine that in the tumor microenvironment, there are a lot of signals there. I mean, TGF-beta is generally thought to be high in tumor microenvironment. So, and I think when we've looked at some of the human sets that others have generated, there does appear to be a TGF-beta signal that's present. But there are certainly going to be other signals that are at play there.
Yes, thank you so much.
All right. Not sure if we have other questions from students. Luisa, are there any questions on your side?
Yeah, we have two questions here.
I'm not sure if he's a student. Do you know, Luisa, if Thiago de Souza Vieira
Hi,
is a student?
Miriam. So I'm actually not a student. I'm a postdoc in the institute. Also a substitute professor of physiology. I got into it because I was interested in mast cells as well. So my question is, I was just curious about how this TGF-beta signaling it's affecting the transcriptome of these mast cells. So I was wondering if you have any insights on how molecularly the TGF-beta is modulating the transcriptome of these cells, and whether they are actually affecting the mast cells that are being recruited or the cells that are already there. Have you done any distinction of this?
Yeah, I mean, that's a good question. So, I guess to kind of break that up into two parts, how is TGF-beta doing what it does? There does appear to be a whole network of transcription factors that are coming on in response to this. And that's going to be one of the things that I think we're going to be trying to look at next, is dissecting how these various transcription factors play with each other. TGF-beta is very good at silencing things, right? It's one of its things that it's well known for, its ability to turn stuff off. So there is certainly something that it is doing to shut down these MCTC associated signals, right? Kinase, cathepsin G basically, drop down to nothing at the transcript level. So, how is that working? We're still trying to think through that. Another possibility is that TGF-beta does have this known capacity to change chromatin structure. So it could be also, in addition to transcription factors, there's probably an epigenetic component to it as well, where it's maybe closing off certain loci, opening up others. But yeah, that's going to be a thing that we're going to be very interested in thinking more about going forward. And then as to your other question about whether it's a signal that's there or whether it's something, is it acting on a cell that's there or is it acting on a recruited cell? We don't have yet the ability to definitively answer that one way or the other. So we can see that there is that this TGF-beta signature goes up in mast cells in the mouse lung when we compare the challenged versus the unchallenged state. So at the very least, the cells that have beta integrin on them seem to be experiencing more of a TGF-beta signal. This could be for a few different reasons. One of the interesting things about TGF-beta is that it is often secreted and deposited in this latent state in the tissue, and it needs to be activated. So one of the things that activates it are a number of integrins that are associated with the epithelium. So alpha V integrin is sort of one of the most well-studied. And there was a study done, I believe the Shepherd lab, maybe 10, 15 years ago. They looked at an alpha V knockout mouse, and when they induced inflammation, they got mast cells in the epithelium, but they all had an MCTC phenotype. So there's probably a component of it that is activated epithelium, turning on TGF-beta. There's probably some component, though, of there being a signal that's there in general. We see in human, in the distal lung, there is a signature that's present. It's not as high as it is in the nasal polyp. But also, we still don't fully understand what mast cell expansion is, right? To what extent these cells are being recruited versus there being local proliferation. So in the mouse, I wouldn't say we have any evidence for local proliferation. I don't, transcriptionally see a proliferation signal that's present. But in human, in single cell sequencing, we absolutely do. So we see that there is this very clear population of Ki67 positive cells, and that's true in nasal polyps. It's true in various human lung diseases. It's true in eosinophilic esophagitis. We saw it in the IBD data set that we looked through. So, to what extent is it a cell that's there proliferating versus a recruited cell seeing a signal? We can't answer that one yet. But it's a very interesting thing that we'd like to know more about.
Okay, sounds good.
Luisa?
Professor Daniel, amazing lecture and data. Thank you so much. So I have a more elementary question, but in the same sense of the Thiago's question. So you have shown that the TGF-beta signaling does not affect the IgA-dependent mast cell degranulation, am I right?
Yeah.
So,
so I should clarify, though, that is true in human, probably not the case in mouse. There's actually a pretty good body of literature in mouse where TGF-beta does inhibit their ability to granulate in response to IgE cross-linking. So, I think it could be the difference between a mast cell that has grown in the signal for an extended period of time and maybe the more acute impact of TGF-beta on the cell.
Yeah. And thinking about the difference between the mast cell populations, did you guys have seen any difference in the expression of IgE receptor, for example, in the two subsets of mast cells?
So, transcriptionally, we were a little underpowered to detect differences. We're working on a follow-up study where we'll be able to dive into that a little bit more. Broadly, I would say, anecdotally, generally, it looks like the recruited ones have a little bit less expression, maybe, than the ones that are sort of constitutively there. I think the wrinkle in everything is that IgE is going way up and IL-4 is going up, and these are both signals that upregulate the high affinity IgE receptor. In humans, we definitely see that there is reduced Fc-epsilon-R1 expression in the intraepithelial mast cells, in nasal polyps. And so TGF-beta alone doesn't do that. So we're not sure if that is due to growth conditions. So when we differentiate mast cells, we grow them in IL-6. And so our ones that we're growing in IL-6 and TGF-beta and stem cell factor, it's possible that the IL-6 is the reason that we see these differences with Fc-epsilon-R1. We don't understand that one yet. But that is, certainly in vivo, we see differences in expression between these compartments. But yeah, it seems like TGF-beta itself doesn't do that, which is another reason we think that, it's just one part of a puzzle. And I think that that's going to be one of the things we really need to understand going forward is, this rich heterogeneity we see in human mast cells is probably the interplay of, dozens of signals. I think they're not dumb cells. They tend to not change their phenotype that much once they're in a place. But, my sense is that they do all the hard work figuring out where they are, kind of integrating local signals and deciding what phenotype to take on. So, I think that's one of the reasons Mina's observation with stem cell factor and TGF-beta was so interesting, right? So TGF-beta itself doesn't really turn on that protease. Stem cell factor does, right? So perhaps TGF-beta tells it where it is if and only if it sees stem cell factor. One licenses the other. So I think that's that interplay we really want to understand a lot more going forward.
Yeah, that's very interesting. Thank you, Professor.
Miriam?
Oh, hi. Thank you very much, Daniel, for the talk. It's really amazing. There's a lot of data. And on this discussion here, I became very curious about that proliferating population. especially after you mentioned that you cannot model that in mice, so it becomes a little bit trickier to do the molecular work maybe. But if I remember correctly, it seemed that it leveled with the intermediate population more than the MC1 or MC3, correct? And
Yeah.
I was wondering if you think that it could be that it is in the process of differentiating that the cells proliferate, or if they would go back to a previous phenotype, proliferate, and then re-differentiate into whatever the new environment could be leading them towards. Would be a final point, or if they could reset their marks, let's say, by entering proliferation and then exiting again.
Yeah, I mean, it's a great question. I mean, certainly when you look at differentiation of, say, T cells, right? Proliferation is a major part of that. There is no better way to open up your chromatin to rearrangement and to being, you know, shuffled around than proliferation. So I like that hypothesis. That's kind of my current pet hypothesis, is that it plays a role in the differentiation process when they're choosing what phenotype to take on. I'm sure that I hope, or hope is the wrong word, but I definitely think that likely the proliferation is a component of that. But it's also possible that there is some pool that's just present in the tissue that has a capacity to proliferate that might be turned on in response to various inflammatory signals that are kind of as yet unknown.
I have a question myself. So I've been working with mouse bone marrow derived mast cells for quite a while. And I was really concerned when your paper in 2016 came out. That's what the hell I have been working with. Are these actual mast cells or not? But seeing your data today, it's interesting how it's actually recapitulates very well the mouse bone marrow mast cells that were cultured on matrigel. That was work done by Rene when he was working with John Arm. And it's actually also was dependent on TGF-beta and increased leukotrienes production. So I was wondering, the mouse bone marrow mast cells are still a valid model. They can be improved somehow by adding TGF-beta or other factors. How do you feel about these particular model?
Yeah, I mean, I think the BMMC is an interesting cell. It's something that doesn't exist in vivo. But when you look at, for example, work done by the Galley lab, when you inject these things into the ear of a mast cell division mice, they will take on a phenotype that is much like an ear mast cell, right? So I think that they're a blank slate that are kind of there to be pushed in different directions by signals. And I think that that's, they're certainly useful in understanding mast cell biology. But I think that the more we can understand how to push them, probably the more useful they'll become. I think it's a tool that we just haven't figured out everything we can do with them yet.
All right, great. I think Dr. Julia Schonstein has a question.
Oh, thank you so much. After hearing your talk, David, I thought that I should actually start the kindergarten for mast cell biology. So because we really need people like you with your expertise to give some, perhaps a chance to have a better insight on our own data. We are coming from a completely different branch of science, which has to do with the microcirculation, plasma leakage, and the connectivity between mast cell degranulation and activation of the kallikrein-kinin cascade. So we are talking about bradykinin release as a function of the integration. You are talking so much about the different levels of integration during an inflammation, and this is exactly what we found. We have 2 papers published. It's all in the hamster cheek pouch, which is a very good model to do intravital microscopy and look at the dynamics of inflammation. And our experience is essentially on T. cruzi infection, which, the Trypanosoma cruzi, it's an intracellular pathogen.
Mhm.
So after a few days, you have a lot of, cell death, injury, and it's incredible how mast cells are connected to the kallikrein-kine system. Because, if you uncouple them, you really control the dynamics of the inflammatory process. Now, the question is, what about polyphosphates? Because, I'm aware of the paper that Roberto do Campo have published several years ago. There was a big controversy because there was a paper in Immunity claiming that heparin was also able to directly activate the kinin cascade. Negatively charged polymers, they have a tendency to activate the contact system, okay? So I'm now convinced that it was an artifact. Heparin doesn't do that because using highly purified heparin, we don't see activation of the system. So it's incredible. It's a paper published in Immunity, but it's probably wrong. However, polyphosphates, it's so difficult to detect, to measure. Have you had any particular experience about polyphosphate? What do you know? Is this, broadly produced in granules or only in certain subsets? What do you know about that? Because that information could help us, to predict in which context you could have the connectivity between this and the kallikrein-kine cascade, which is it's a very powerful system for propagating inflammatory reactions. Okay, so this is one question that I had. The second one, maybe we can leave it for another time because I think that you are already tired. So after so many discussions, so.
Well, unfortunately, I have a short answer for you, which is I know nothing about this system in these cells, but I will definitely look it up now and see if there's anything I can pull out. I mean, this is one of
maybe I can try the second question. If Bruno gives me the opportunity, I don't know if the time is over or not. I have still a chance or not.
Actually, I just want to go to another question. Then we can come back to the second question,
good.
if you don't mind. Thank you very much.
No.
Cristiane.
Hi, beautiful body of data, Daniel. Thank you very much. I was wondering, and it's kind of a question or a suggestion. When you were characterizing the subsets and looking for difference between them, have you or Mina looked at ultrastructural level? I mean, based on your data of enhanced eicosanoid formation, production of CSLT and PDD2, and because your main stimuli is TGF-beta that I know induce beautiful biogenesis. When I'm asking you about ultrastructural difference between your subsets, I'm particularly interested on lipid bodies or lipid droplets. What can you tell me?
We haven't looked. It is something that is on the to-do list. We kind of briefly looked into it and realized that it was something that we needed to actually get some grant funding for if we were going to do. It was going to be a little bit beyond the budget we had for the project. But I think that it is something that is going to certainly be going into a future grant because I think there's certainly like the mast cell has a beautiful ultrastructure and all the differences across different compartments. To whatever degree we could recapitulate that in vitro would be very interesting to understand. So certainly would love to do that. Need to get the money first.
Okay. Yeah. Thank you.
Hey, Julia, your second question, please.
Second question has to do with cardiac mast cells. For other reasons, because we have this interest in the Chagas disease cardiomyopathy, right? So we ended up reading a few reviews about cardiac mast cells, and it called my attention that chymase from humans and hamsters, they have the same substance specificity in the sense that, to my surprise, both are able to generate angiotensin II independently of angiotensin converting enzyme. And angiotensin 2, as you know, is it's pro-angiogenic, pro-fibrotic, hypertensive. It's a bad, supposed to be a bad boy. So I ask you, it was blew my mind when I saw your data showing that TGF-beta is actually inhibiting the production of kinase. Is this a general, or do you think that maybe there is still room for exceptions, so that, let's say, in the cardiac scenario of inflammation, we may have both TGF-beta and angiotensin 2 generated by kinases from mast cells, acting in synergy in a pro-fibrotic process. So have you ever, come into these thoughts,
Yeah. So I think one of the things that, and I am now kind of wishing I sort of expanded on this a little bit more. One of the things that I find so fascinating about TGF-beta is that it has the mast cell plasticity appears. There's like sort of two worlds. One is what it can do to surface proteins, right? And to their ability to make lipid mediators. And that's very dramatic. 7 days, you get it. And the other is the granule phenotype, right? So even though treating them with TGF-beta knocks the kinase transcript expression down to 0, it does not, the expression within the cell does not budge. So I think that this has some interesting implications, right? Because when we look for mast cells in tissues, we do kinase staining or tryptase staining. But what our data suggests is that is telling you the granule phenotype of the cell, sure, but it might not have a one-to-one link with the effector program. So it could be that, for example, in inflammation resolution, when you have TGF-beta within the tissue, the mast cell is still, even though the kinase expression within the cell might be going down, it still has plenty of kinase in its granules, but also maybe gaining this ability to make more cystoleucotrienes and more PGD2. So that's sort of one aspect of it.
That's a good point. Could be a biphasic process, right? Does not interfere with the granular function of the kinase, but it's actually trying to control so that you don't have exaggerated responses, and maybe allowing for resolution coming later on. That's a good point. Thank you. Thank you.
All right. So I guess I'll make the last question. Is about specifically the lipid mediators that are being produced by these different types of mast cells. And one thing that I noticed, one was the how COX-1 was supposed to be the central cyclooxygenase protein, although cyclooxygenase 2 seems to be expressed as well. So maybe there is some sort of subcellular specific location that lends to this type of dependency. And the other thing is, I saw as well that leukotriene for hydrolase was also had its expression modulated. How was LTB4 production on these cells?
Well, I guess to answer those questions in reverse, yeah, we haven't looked for LTB4. So presumably there would be more because the enzyme is there, but we haven't done the experiment. So that is certainly something that we'd like to understand a bit better. But the COX-1, COX-2, I think it's a very interesting sort of system, right? Because generally we think of COX-2 as something that can be induced by inflammation, and COX-1 is more stable. So, the way I'm thinking of it at the moment, which is very well maybe completely wrong, is that when we look in vivo in a nasal polyp, there is plenty of COX-2 expression. In vitro, we don't see a ton of COX-2 expression. So when we're activating the cells and we're looking at lipid mediators, we're looking a couple hours after we've done IgE cross-linking. Generally, we think of COX-2 as something that maybe requires about 24 hours to turn on. So my thought is that the increased COX-1 expression puts the MCT, or the intraepithelial mast cell, at a higher baseline ability to make PhD2. But inflammation can upregulate COX-2, and it might upregulate it more within the subepithelial mast cell or the connective tissue mast cell, such that ultimately, their ability to make these things is going to be a cross product of how much COX-1 and COX-2 they have. Certainly, there could also be differences in subcellular localization, but I think that it's almost as if the COX-1 sort of sets your floor, and then the COX-2 is more the ceiling. So I think that perhaps the floor for PhD2 is much higher in an intraepithelial mast cell, but that doesn't necessarily mean that the ceiling is higher.
Right. Well, and it also has, I think, something to do with the type of stimulus that you're being used in. That the IgE triggering is very strong, and it can actually drive lipid-mediated production through COX-1. But if you, I don't know if there is any model, such as in the mouse mast cell, that for delayed prostaglandin D2 production that is exclusively dependent on COX-2, although COX-1 is also present there. But since COX-1 depends on a higher peroxide tolerance inside of the cell, you actually only have COX-2 that is active in these kind of settings. But yeah. I actually have I'm sorry, we are running out of time. I don't want to delay your lunch too much. But there is one other question: is how essential is the epithelium in the differentiation of the mast cells in the tissues? These TGF-beta could be coming from another cell source, and since Cristiane is here, could it be coming from eosinophils, for example?
It certainly could. That's something that we haven't really looked into, partly because of the technology that we tend to be using, right? So single cell sequencing is great at seeing almost everything that's not an eosinophil or a neutrophil. Those TIL cells are almost impossible to detect through single cell sequencing, just for reasons that are still not totally clear, but probably have something to do with all of the endogenous RNases that are present within the eosinophil. So we don't have a great sense of that. One thing is that in aspirin sensitive nasal polyposis, there's a lot of eosinophilia. There's also a lot of mast cells that have the intraepithelial mast cell phenotype. There's, I would say in that, something like 90% of the mast cells have an intraepithelial phenotype. In aspirin tolerant nasal polyposis, maybe it's more like 60 or 70% have the intraepithelial mast cell phenotype. So it certainly could be that when you get a lot of eosinophilia in a tissue, it could be amplifying the system. And in fact, it does look like in aspirin sensitive nasal polyps, there's a higher TGF-beta signature score. So when we look at those violin plots, it seems to be higher in that disease than in aspirin tolerant. Eosinophilic esophagitis is another place where you get a lot of eosinophils and a lot of mast cells. And the mast cells that expand are preferentially in that intraepithelial space. So it certainly could be that the TGF-beta can come from multiple places. I think, as far as structural cells go, the epithelium is sort of the structural cell that seems to have the highest expression. And certainly of the cells that we can detect through single cell sequencing, it appears to be a dominant source. But yeah, EOs are invisible as far as single cell sequencing goes, so we don't have a great read on that.
All right. So I think the take-home message from this talk is that mast cells are cool, eosinophils are a pain in the ass. So we can just take whatever we want from this. No
comment on that one.
No, kidding aside. It was a really great talk. Thank you very much. Next week, we are going to have another very nice talk from Dr. Jessica Lancaster from Mayo Clinic, age-associated change to lymph node stroma cells, impaired T cell homeostasis and function. So I think we are really on a roll here with very nice talks. And Dan, thank you very much. It was really wonderful to hear someone that actually has some very interesting data on mast cells. Thank you.
Thank you so much again for the invitation. This was great. Fantastic discussion.
All right. Thank you.
Thank you very much.